So good evening, everyone. We're very pleased this evening to have Dr. Richard Gornall from the University of Leicester with us. Richard is director of the University Botanic Garden in Leicester, and he's going to talk to us this evening about saprophytes in the British flora. So I think we're going to hear about fungi. I think we're going to hear about orchids. I think we're going to hear about the evolution of carbon acquisition. So with all that to get through, I think I should hand over to Richard and invite him to start his talk. Richard.
Okay, thanks very much, Alan. Thanks everybody for inviting me to give this talk. It's the first one I've ever done under these circumstances, so I hope it turns out all right. The inspiration for the talk actually comes from discoveries which were made by my colleague Isabel Raval near Melton last year and this year. Last year she discovered Pyrola rotundifolia growing in some dark woodland, and this year she backed that up with the discovery of Hyperpitis nearby. These of course are exciting discoveries. This is 2 new native species to the Leicestershire flora. And it prompted us to start looking in the literature about these species to see if we could get a better understanding of how they got here. Now, when you look in the literature, they are credited with being saprophytes. Now, it's important to understand what a saprophyte actually is, because the question mark in my title, at the end of my title, is very important. So please, Alan, don't broadcast it without the question mark in the title. So what is a saprophyte? Well, I had a conversation about this in my younger days when I was out in the field with Professor David Webb, and we were looking at a bird's nest orchid, and I was quizzing him on how it got its mission. And he said, it's a saprophyte, dear. So I said, okay, I couldn't really quite get to grips with how a flowering plant could be a saprophyte. So I asked him in a bit more detail, and the best he could come up with was that he said, saprophytes eat dead things. And so I didn't want to pursue it, I left it there. But he was right, of course, saprophytes do dead things. And good examples are bacteria, fungi like Rhizopus species, that's bread mold, and in the aquatic environment, water molds, which are oomycetes. These are not true fungi, of course, they're actually more related to brown algae. But nevertheless, they are molds. They sort of behave in a similar way to fungi, and they extract their carbon from dead organic matter. Now, in the British flora, if you look at the reputable literature, so the standard floras and so forth, so books like for those of you who've been in botany for some time, Clapham, Tooten and Warburg, or Clapham, Tooten and Moore. Recently, volumes by Stace and indeed Poland and Clement, the vegetative key to the British flora. If you look in the new atlas of the British flora, and indeed all the wildflower guides that you might happen to come across and use in the field, all of these claim that some plants that lack chlorophyll are saprophytes. And I've sort of partial list on the right-hand side there, which I'll go through briefly in a minute. But basically, things which are claimed to be saprophytes seem to be any plant lacks chlorophyll that is also not parasitic. So non-parasitic, non-photosynthetic plants have had this blanket epithet put on of saprophytes. And some examples which have been claimed to be saprophytes are a couple of mosses, Buxbaumia aphylla, and Sphagnum sphaericum, we'll come to those in a minute. Some lycopod gametophytes, these lack chlorophyll, they're not parasitic, so they've been claimed to be saprophytes. Some ferns, in particular fern gametophytes, adder's tongue fern, these plants lack also potential saprophytes, which are the flowering which people are more perhaps familiar with, if only through pictures. So the ghost orchid, Epipogium aphylla, the wintergreen species, Hyperpitis monotropa, sorry, Dutchman's pipe, Hyperpitis monotropa, bird's nest orchid, Neotia ledicebus, and coral root orchid, Mycorrhizae. All of these lack chlorophyll, or at least have very, very, very little of it. And these are all claimed in the literature, much of it the recent literature, to be saprophytes. Now, they're called saprophytes because they don't qualify as parasites. Now, to qualify as parasites, typically there is an identifiable connection between the parasitic plant and its host. So, for example, toothwort, if you dig down in the soil, you can find a connection between the rootstock of the Lathria, the toothwort, and its host. You can act as a physical, which looks a bit like a root, which goes from to the host. Ditto with the branch broom rape, and you can see pictures down at the bottom right, that red illustration. There's a labeled Horstorium. This is produced by the parasitic plant, and it plugs itself into the root or the rhizome of the host plant, and it sucks the carbon out from the host plant. On the right-hand side, I've just put for fun the sporophyte, or the spore-bearing, the cone-bearing stems of Equisetum arvense. This is a heterophytic species. In other words, the vegetative stems look very, very different from the reproductive stems, and these reproductive stems have chlorophyll. But if you dig down in the soil, often quite a long way with Equisetum, you will find these colourless stems connected into the ordinary rhizome network that exists. So these plants are all parasitic, either on their hosts, or in the case of the Equisetum, it's parasitic on itself, if that's a concept you can bear to have. So saprophytes then are none of these because they lack these connections, these obvious connections with the host. So some of the plants which have been claimed to be saprophytes include, as I mentioned earlier, the Buxbaumia. Now this is the little thing on the left, the bryophyte on the left there. It's often described as being achlorophyllous, and therefore potentially saprophytic or heterotrophic, but it's not true. Despite its reduced habit, all Buxbaumia species apparently contain chlorophyll, so they are autotrophic. The Splachnum, which is another moss, that has wrongly been claimed to be saprophytic because it actually has green gametophytes, which you can see in the picture there on the right hand side. So if you look down here, you can see the green gametophytes. This is the dung, which grows on, and it's because it grows on dung that it got this reputation for being a saprophyte. It isn't a saprophyte. It gametophytes just grow in the normal way. They just happen to have dung as a habitat. But what about these? These are a bit more likely. So the ghostwort, which is a little species of liverwort on the left-hand side here, these lack chlorophyll, both in the gametophyte stage and in the sporophyte stage. And these are prime candidates for being saprophytes. Analogous to the Equisetum example, we have some lycopods where the gametophytes don't have any chlorophyll. So the question is, where do they get their carbon from? And similarly with the some of the orchid genera, in particular in the Ophioglossaceae, the adder's tongue orchid group, these gametophytes, this is a gametophyte of the adder's tongue of the moonwort, Batricium. This gametophyte lacks chlorophyll, and therefore the question is, well, how does that gametophyte get its nutrition? Is it saprophytic? In terms of flowering plants, there are perhaps more examples of species which have been known to be saprophytes. So we've got the ghost orchid here without chlorophyll, bird's nest, coral root orchid, sometimes lack completely, sometimes it seems to have a very little bit in it. This is a very pale green form, so this obviously has a little bit of chlorophyll. And then the Dutchman's pipe, Pittis monotropha, again without chlorophyll. So these are all candidates for being saprophytes, and in the literature you will see that they are indeed claimed as being saprophytes. Well, what's the evidence that we've got here? Why aren't they parasites? Well, they're not parasites because they don't have these underground connections. When you dig down in the ground around these species, from very relatively early on in the 19th century, there were frequent reports of thread-like filaments which were associated with the stubby root-like structures. Clearly, these stubby little root-like structures, or these stubby roots, did not, but never were associated with some thread-like filaments. But other than these thread-like filaments, whose ends you couldn't possibly find. Yeah.
Richard, sorry to interrupt. Your sound isn't very good. I'm going to just try. I wonder if you could turn your video off. Don't turn your microphone off. Just turn your video off, and see if that improves the sound.
Okay. If I turn the video off, you might lose screen.
Yeah. Well, we'll find out whether that happens or not.
Okay. Hold on. Stop video.
Okay, and now if you put your slides on full screen. Okay, yeah, back to you again. Sorry for the interruption.
The screen so that the microphone picks up more. I can turn my sound up a bit, although that might not help if it's crackly.
Yeah, it's cutting in and out a bit. So
okay.
All
right. Well, I'll go a little slower, and interrupt me again if it gets impossible.
Okay, thank you.
All right. So early digging around the base of these potential saprophytes revealed some little thread-like filaments, but where they went, nobody really knew. In the 1840s, they were identified as fungal hyphae, so people did from quite early on recognize that these were fungal filaments. And it wasn't long, 40 years, before they were actually demonstrated to be mycorrhizae. So we knew that mycorrhizae infect these potential saprophytes, but more than that wasn't really known. But nevertheless, despite realizing mycorrhizae were associated with the roots, these achlorophyllous plants continued to be regarded as saprophytes, and it believed they got their nutrition from dead organic matter. Yeah. Now, if you are going to be a saprophyte, you need a battery of enzymes to extract the carbon. The enzymes that you need include cellulosases to break down cellulose, turn into glucose. Importantly, you need enzymes to break down lignin, which will turn it into simple water-soluble compounds. You need amylases to break down starch, proteases to break down proteins, and lipases to break down lipids to turn those into fatty acids and glycerol. However, I won't bore you with details, but all of those candidate saprophytes that I've mentioned, none of those that have been investigated turn out to have the necessary enzymes. So clearly there's a big question mark, is if they don't have the enzyme machinery, how do they get hold of carbon from dead organic matter? Well, the truth of the matter is, of course, they can't, and they have to get their organic matter some other way. And this is where the mycorrhizae come in. It wasn't until 1960 that the critical definitive experiment to demonstrate the role of the mycorrhizae was conducted. It was done by a Swedish botanist called Eric Björkman. And what he did was he located populations of monotropa in various woods around where he lived in southern Sweden. And what he did was he selected at random a group of plants, a group of individuals over which he put stainless steel cylinders, and the cylinders down about 50 centimeters in. So each of these Dutchman's pipes enclosed with a cylinder, quite a wide diameter, so that you didn't damage the plant. But these cylinders were plunged down over the Dutchman's pipe individual. So each one had a sort of steel sleeve, if you like. Now, the idea of this, of course, was to break any with any plant. And after about a year, he went back to look at his plants and his control groups. And what he found was that the plants without cylinders were vigorous and produced new shoots. And the plants that had got cylinders were weak and had developed no new shoots at all. Now, of course, by itself, this doesn't mean terribly much, because you could imagine that there might be a number of causes for a plant being weak if it's lived for a year inside even an open-topped cylinder. But nevertheless, that was the one thing he did to demonstrate that these did not survive very well in the absence of a connection elsewhere. The crucial thing he did was to inject carbon and phosphorus 32, these are isotopes of carbon and phosphorus, into the neighbouring pine and spruce trees. Now, pine and spruce are autogreen, they photosynthesize, they produce their own carbon from the atmosphere. Now, what he did effectively was to radio label this carbon that the trees had fixed from the atmosphere, and he then measured or assayed the Dutchman's pipes for the radioisotopes, the radiocarbon and the radiophosphorus. And what he found was that nearby plants, that is plants nearer the trees, had high levels of the isotope, and the further away from a tree you went, so the levels of isotope reduced. And so at a long distance away, the Dutchman's had no isotope. So the first time was a very clear demonstration carbon had travelled from a tree to a Hypophytis individual. And this was a radical demonstration of the function of these Mycorrhizae, which had been known about for some time, but whose function in this particular context hadn't been fully appreciated. So clearly the Hypophytis was getting its carbon from the trees through these Mycorrhizae. His third experiment to support all these results was to do with isolating the mycorrhizal fungus from the Hypophytis. And he showed that this fungus, even though he didn't know quite what it was, what species it was or anything, he showed that it could also form mycorrhizae with the pines and spruces. So by these three experiments, what Eric Björkman had done was to demonstrate that Hypophytis monotropa was not saprophytic, but instead was parasitic on a mycorrhizal fungus. And this was the first case of proven what is now called mycoheterotrophy. So here's just a little schematic to show you what goes on here. So you've got your tree, which fixes carbon dioxide from the atmosphere. The mycorrhizae, which are associated with the tree roots, various typically they're ectomycorrhizae, but we'll come on to that in a minute. The mycorrhizae extract carbon from the tree and give back to the tree nitrogen and various other minerals. So there's an exchange here. In turn, the Hypophytis monotropa derives its carbon from the mycorrhiza and nitrogen and minerals from the mycorrhiza. So the Hypophytis is actually parasitic on the mycorrhiza. So it's not a saprophyte, it is in fact a parasite, but a rather strange one. And mycorrhizae has now been demonstrated in a number of different places around the angiosperm evolutionary tree. And as far as we know, at the moment, it's evolved at least 7 times in dicots. That includes Dutchman's pipe, the heather family, for example. So at least 7 times in the dicots and 40 times in the monocots, 30 of which have been in the Orchidaceae family. So it's a bit of a niche mechanism for getting your nutrients, but less it has happened more than once, and it is reasonably widespread. Now, obviously, the Mycorrhizae are important in all this. They're the kind of middleman. And so it's helpful to understand a little bit about the mycorrhizal fungus, which is acting as an intermediary between the tree, which is the source of the carbon, and the hypophytis, which is the ultimate sink of some of that carbon. Now, this is a little diagram, which is basically a cross-section through a root. You can see there are different types of mycorrhizae that are illustrated here. There isn't just one type of mycorrhiza. There's at least 6 or 7 types, but there's a basic 5 which are illustrated here. The commonest is the arbuscular endomycorrhiza. I apologize, well, I shouldn't apologize. They're rather long words, but that's what they're called. So an arbuscular mycorrhiza is believed to be the ancestral condition because it's been found in Cooksonia fossils, which are 400 million years old. So these are some of the earliest vascular plants. So it's believed to have a long evolutionary history. These arbuscular mycorrhizae they penetrate the cells, they occupy the inside of the cells, as is illustrated here, where they form either little branch structures called arbuscules or balloon-like structures called vesicles. So something like 85% of all vascular plants have got these arbuscular endomycorrhiza. So this is the common condition, and it's the ancestral condition. Now, there are variants, slightly different types of endomycorrhiza, which occur chiefly in the orchid family and in the heather family. In the orchid family, the endomycorrhiza form these coil-like structures inside the cell. So they're not branched or balloons, as in the arbuscular endomycorrhizae. They're coil-like structures. And something similar happens in the heather family with these ericoid endomycorrhizae. They also form coils of a slightly different nature, but nevertheless they are coils. Cells. So these 3 types are in fact inside the cells. The 2 other main types are external to the cells and occupy either a sheath around the outside of the root and or occupy spaces between the cells inside the root. The commonest type is just called an ectomycorrhiza, which you can see in pink at the top of the screen there. Typically, you've got this fungal sheath surrounding the root there, with the filaments going off into the substrate, into the soil. This fungal sheath actually proliferates down between the cells towards the center of the root to form a sort of network of fungal hyphae between the cells, but within. This is called a Hartig net, presumably after Hartig. I'm afraid I don't know who he was. Maybe Jeffrey will know, I don't know. Don't want to drop him in it though. Right. So these typically are found on big trees, or in the northern hemisphere at least. Their prevalence in the tropics is not well known, so I'm not sure if that's a correlation or not yet. But this is a relatively common-ish type of mycorrhiza, and something like 10% or so of plants have this kind of mycorrhiza, compared with 80% having the arbuscular type. Then there's The fifth kind of mycorrhiza, called the arbutoid mycorrhiza. Now, this is like ectomycorrhiza in that it forms a sheath around the outside of the root, but that sheath typically does not form a heartic net extending deep in towards the center of the root. It occupies the outer and perhaps the epidermal layer of the root in between cells. A particular kind of arbutoid mycorrhiza, the fungal hyphae do penetrate walls, making little peg-like structures, as you can see here. So you can see from this that the orchids are a bit different with their mycorrhizae, their own special sort, but the family is also different. And in particular, it's more than any other family. So we have heather type of mycorrhizal structure here, which occurs almost throughout the heather family. And in one particular part of the heather family, we have these arbutoid sorts. In it, these rise not just things like arbutus and Arctostaphylos, but also pyrola and hypophytis. Anyway, there's your mycorrhizae. Are very common, so common in fact that it's the exception to find a plant which doesn't have a mycorrhizal association, and this has led to the concept of what's been called the wide web. Basically, everything in a landscape is linked by mycorrhizae. It's quite an impressive concept when you stop to think about it. You look out over a landscape and you sort of look at all the vegetation and realize all this stuff is connected. There's just a massive connections under the ground there, linking one plant with another. Now, if we look at the fungal partners, for a long time, because fruiting body alone, it was a mystery as to what the nature of the fungi actually were. But now with the advent of DNA sequencing, we've been able to pinpoint the identity of many of these mycorrhizae. The commonest mycorrhiza, that is the which occurs in about 85% of families, they have a particular class of fungi associated with them called the glomeromycetes. Now, these weren't properly recognized until relatively recently. They are, in fact, a class of fungi which is sister to the ascomycetes and the basidiomycetes. So if you have that pair, ascomycetes and basidiomycetes, which is to each other, sister to that pair are the glomeromycetes. And these are the fungi which make up almost all of the mycetes which you will find in nature. Mycorrhizae comprise up to 10% of the plants. They are made up of zygomycetes, ascomycetes, basidiomycetes, and there may be one or two other classes of fungi as well. There's a whole mixture there. When we drill down into the orchids, what we find is we're dealing with basically basidiomycetes and ascomycetes are the more common mycorrhizae, common ascomycetes. With the headers, the Ericaceae endomycetes, seeds and common fungal associate. When we get into the arbutoids sorts, which is that these are the ones with the Dutchman's pipes, for example, the hypophysis, these are basidiomycetes. Now, it has been claimed that mycorrhizae are absent from mosses. I've got that on the screen as a fact. This is debated in actual fact. Mosses are actually, many mosses, not most, but there are many, a significant minority, let's put it that way, a significant minority of mosses are associated with with one fungus or another, but it's debated as to whether the association qualifies as a mycorrhizal association. So that statement at the bottom there needs a bit of qualification. Similarly, most liverworts do not have a recognized official, as it were, mycorrhizae, except for one, And I showed you a picture of that species earlier, the Anura. Hornworts also don't appear to have a proper mycorrhizal association, although they do have some fungi which live on or near the roots, but they don't qualify at the moment as mycorrhizae. A lot more work needs to be done on these groups which apparently lack mycorrhizae. It may well turn out that these do have mycorrhizae, but there'll be additional types. There's a few angiosperm families, particularly aquatic families, from which mycorrhizae have not been reported. And it's certainly true that even in a family which does have mycorrhizae, it's not necessarily true that all the species have a mycorrhizal fungus associated with them. But by and large, as a rule of thumb, if you just gaze out at a landscape, you can be assured that almost everything that you see will have a mycorrhizal associate. Here's the picture in the heather family. Now, there's a lot of generic names here which you probably won't recognize, but I have starred, I've asterisked all the ones which are British or Irish genera. So there's Hyperpitis, Arbutoides, that's a subfamily of the heather family. So Arbutus, the strawberry tree, and Arctostaphylos, the bearberry, they're in here. Orphilia is in here, Pyrola, and Monastees. These are green. These are photosynthetic species. All of this lot here qualify as having the arbutoid type of mycorrhiza, although the monotropes, that's this lot here, Sarcodes upwards, have a particular variant of the arbutoid sort. The rest of the heather family has the ericoid type of mycorrhiza, and the basal genus in the heather family, Enkianthus, has the common arbuscular mycorrhizal association with a glomeromycete. So you can see you've got the glomeromycetes, then you've got the ericoid mycorrhizae, and then all of this lot has the arbutoid type of mycorrhizae. And you can see that there is an element of specialization. So, for example, Pitopus and Hyperpitis, which are sister genera, only have Trichiloma species as their fungal partners. Those Trichiloma species are, the genus is shared with Allotropa, but Allotropa has different Trichilomas. Similarly, Hemitomes has species of Hydnella, none of the others do. Pluricospora has Gaultiera, for example. Pterospora and Sarcodes has Rhizopodon. So there is an element of specialization, an association between a particular genus of monotrope and a particular genus of, or family in this particular case, of fungal partner. So unlike with the glomeromycetes, where the relationships are relatively non-specific, once you get into the monotropoidy, you get increasing level of specialization. Right, sorry, can I check that I moved on? Yes, okay. Right, so where do we go from here then? Is that the end of the story? We've debunked the idea that we've got saprophytic species. Instead, we've got mycoheterotrophs. Well, people continued to batter away at this question to find out more and more about the nature of the relationship. And one set of experiments and studies conducted by Hinson and so forth, and et al., not so long ago, 10, 15 years ago, utilized the fact that isotope ratios between naturally occurring carbon-13 and carbon-12 and naturally occurring nitrogen isotope 15 versus 14, they discovered that these levels were elevated in fungi compared with autotrophic plants. All right, so in other words, fungi have a greater ratio, a greater proportion of carbon-13 and a greater proportion of nitrogen-15 in their cells than autotrophic plants do. And so Hinson et al. Thought this would be a good way of finding out to what extent this mycoheterotrophic nutrition was occurred in other species of, for example, the heather family or the orchid family. So what they did was they investigated a whole bunch of species that lacked chlorophyll, and those are in the purple down here. So they looked at the coral root orchid, the bird's nest orchid, and the Hyperpithis, the Dutchman's pipe. These are just British examples. They looked at examples from other countries as well, but I just put the British species in. These were known to be mycotrophic. And when they investigated the levels of carbon-13 and the levels of carbon-15 in these plants and compared them with autotrophic species, which are in this graph here. So here's the autotrophs. These are green-leaved species, and set to that they don't elevated carbon-13, they don't have elevated nitrogen-15. So this is your baseline, your reference point, or your control group, if you like. Compared with that group, you can see that mycoheterotrophic species have increased levels of nitrogen-15 carbon-13, much you might expect, given that these plants are parasitic on a fungus, even though it is a mycorrhizal fungus. Now, the interesting thing of part of this study came when they included in it some evergreen species which are related to some of these mycotrophic species. So in this particular case that I've illustrated on the graph here, we've got Hyperpithis monotropa, which is in the heather family, and not too distant from that species, if you look on the evolutionary tree a couple of slides back, you'll see Cumaphala and Pyrola. So these two genera are quite closely related to the Hyperpithis. And question is, do they have any sort of hint that they might be on their way to being mycoheterotrophs? Well, the answer to that is yes, they do, but. So this is where the evergreen species fall, with the blue line, the evergreen species related to the Hyperpithis. This is where they fall. And what you can see is that they are elevated in terms of nitrogen-15, which has clearly come from the fungus. So from their mycorrhizae attachment, they'll have got this. But they are not elevated in terms of the amount of carbon, in terms of the type of carbon they've got. So these plants are not extracting carbon from their mycorrhizal host, but they are extracting nitrogen. And you could argue that this is just simply a very efficient mycorrhizal association going on. Nevertheless, people have decided that these plants, Cumaphala and Pyrola, are partial mycoheterotrophs on the grounds that they are abstracting elevated levels of nitrogen-15. But it seems to me that that's all that's happening is that they've just got a very efficient mycorrhiza going on. We don't talk about these autotrophic species as being parasitic on the mycorrhizal. These are exchanging carbon or they're donating carbon in exchange for nitrogen minerals in the same way that these are. It's just that these are extracting the nitrogen much more efficiently. So there is a continuum here between what you might call a balanced relationship between the host and the parasite, and something a bit more one-sided, right? But I think it's legitimate to call these things partial mycoheterotrophs because at the seedling stage, they certainly do abstract carbon from the autotroph. These measurements are taken on adult plants, but if you look at seedlings of these two things would be over here in the purple sector. So at the seedling stage, these are parasites on mycorrhizae. They do abstract carbon from them. So on those grounds, I think it is legitimate to call them partial mycoheterotrophs. And if you don't want to get your tongue around that long phrase, then mixotrophic is the word to use. Okay. In other studies, Orphilia and one Pyrola species do have elevated levels of carbon. So it's clearly a timing issue. If you're mycoheterotrophic at the seedling stage, how long is it before you can live on your own without any help? And clearly some species, in this particular case, the Chimaphila and the Pyrola that was used here, once they're adult, they don't need the carbon support of the mycorrhiza. But some species, clearly even at an adult stage, do continue to need the support of carbon from the mycorrhiza relationship. Okay, I hope that wasn't too garbled, but can you see that we've got three things here? We've got you've got your baseline standard control autotrophic species. These are your green plants, nothing special about them. They do have mycorrhizal associations, but that's where they are. No elevated levels of carbon-13 or nitrogen-15 compared with these other plants. These other plants do have elevated levels, and these ones in particular, it's because these are parasitic on the mycorrhiza. It's debatable whether you regard these as parasitic on the mycorrhiza, since they're not abstracting carbon. One or two of the species do abstract carbon, so those clearly would be regarded as parasites. But these ones here shown in the graph, I would argue were just simply very efficient at abstracting nitrogen from the mycorrhiza. So there's no carbon transfer there. So on those grounds, I wouldn't regard these as parasitic. Now, this whole idea of partial mycoheterotrophy began to take off, especially with this use of carbon-13 and nitrogen-15 as a sort of indicator as to whether you were getting more than your fair share of fungal compounds. And in a survey of meadow orchids, these lack carbon-13 enrichment. So in other words, they would fall in this blue circle here, because they lack the enriched carbon-13, but they do show enriched nitrogen-15. So these meadow orchids, so for example, green-winged orchid, various marsh orchids, they would probably all locate themselves in this blue circle here. They're getting, you know, a good amount of stuff from the mycorrhiza, more than the autotrophs are, but they're not getting any carbon. Nevertheless, in the literature, many of these meadow orchids have been branded as being partially mycoheterotrophic. I put that in quotes. It's not quite the right term for it. They're not getting carbon. On the other hand, the situation does seem to be more complicated than that, because in another species, Pseudorchis albida, in the adult stage, it doesn't show enrichment of any sort whatsoever. And yet it does have it seems to have a standard orchid mycorrhizal association. So clearly there's more going on here than we actually are fully aware of at the moment. So it's be prepared for this picture to get a bit more complicated, and by that I mean a bit more interesting. So just to summarize the nature of the relationships, we've got the tree fixing the carbon dioxide, which is then abstracted by its mycorrhiza, and in return the mycorrhiza gives nitrogen and minerals back to the tree. So this is a mutualistic relationship, but as I mentioned earlier, it's a continuum. The balance about how much carbon you extract and how much nitrogen you give back varies hugely, and it can be a rather one-sided or lopsided relationship. But basically you can regard this as mutualistic. Both partners benefit in some way. Over here, we've got our mycoheterotroph, the parasite, which extracts carbon from the mycorrhiza and nitrogen and minerals. No doubt about it, this is a parasitic relationship. Mycoheterotrophic, parasitic. And then we have these mixotrophic plants. These are green, so they do, in the adult stage at any rate, they do their own photosynthesis and fix their own carbon. Nevertheless, at the seedling stage, they will take carbon from the mycorrhiza, carbon and nitrogen and so forth. So at the seedling stage at any rate, they are parasitic, but in the adult stage, they make their own carbon. So on those grounds, you could regard these as partial mycoheterotrophs, in other words, mixotrophic. It is unknown whether carbon is returned from the adult to the mycorrhizal fungus. We don't know if that happens or not. So it's not sure, we're not sure whether this is a mutualistic relationship or an early parasitic relationship. By early, I mean happening at a juvenile stage. So you'd think that would be the end of it, but no, people have still been investigating these relationships and have turned up an additional interesting piece of or set of observations. And this work was done in Japan by a Japanese botanist called Kenji Suetsugu. These are relatively recent experiments, just published this year. And what Suetsugu was interested in was, he said, okay, if we have these mycoparasites, they're sitting there in the ground, they've attached themselves to a mycorrhizal fungus through which they're getting carbon from nearby trees. Have they attached themselves to anything else? Or indeed are there species which attach themselves to things which are not typical mycorrhizal fungi? And what he did was to make use of the fact that the amount of carbon-14 present in the atmosphere increased as a spike in the late 1950s, peaking in the early 1960s. Now, this carbon-14 spike, which you can see in the diagram on the left there, was produced by all the atomic weapon tests which took place in the Pacific, around the Pacific in Australia and in, I'm not sure where the Russians did theirs, presumably in Central Asia. Anyway, there's a lot of atomic weapon testing going on, which meant that the amount of carbon-14, naturally occurring carbon-14 in the atmosphere, increased to the point where it's not natural anymore. This is all man-induced. So the massive spike in the amount of carbon-14. Then the nuclear test ban treaty coming into force in the early 1960s, the amount of carbon-14 in the atmosphere declined. And right now it's almost back to where almost back to what you might call natural levels. Now, what this means is that if you look at some dead trees of, let's say, 20 years old, 30 years old, 40 years old, 50 years old, 60 years old, all right, you can see where I'm going with this. The older the dead tree, the more carbon-14 there will be in its wood. And this means you can actually test whether fungi have actually acquired any of this carbon-14 from really old trees, really old dead trees. And what Suetsugu did was to look at a range of species without chlorophyll. So these are all mycoheterotrophs. So pyrola is a partial mycoheterotroph. Bird's nest orchid, partial mycoheterotroph in Cephalanthera. This is a mycoheterotroph, and so are these black chlorophyll. So there's a range of orchids which are in red there, and a couple of heather family members which are in the purple there. And what he did was to look at the amount of carbon-14 in these plants. And at the same time, what he did was isolate from the roots of these the fungi which were forming mycorrhizal connections with the plant. So in Monotropastrum, he isolated a species belonging to Russulaceae. Ditto with pyrola, these are Basidiomycetes. And then in the bird's nest orchid, he isolated a member of the Sibacinaceae, and so forth. And you can see that for most of these things which have been regarded as mycoheterotrophs, the mycorrhizal associate is an ectomycorrhiza, right? So these are of one sort or another, either an arbutoid sort or a typical ectomycorrhiza sort. In these other orchids down here, Certosia, Gastrodia, incidentally that pretty picture there is a Gastrodia species, and Eulophiat nuania, these orchids down here were associated, perhaps surprisingly, with wood rotting or leaf litter decaying fungi, saprophytes. So here we have Armillaria in these two orchid species, Sathirella in that orchid, and Phlebia in that orchid. These are saprophytic species, not normally regarded as being mycorrhizal fungi. Right, where are we going with this? Well, what we've got then is a range of carbon-14 dates for these species here, plus his autotrophic controls. These are just green plants, and the fungal associates, some are ectomycorrhizal, some are arbuscular mycorrhizal. So those are his controls. So he's got a range of carbon-14 measurements in all these plants, and he's got some species identifications regarding the fungal associate. And then just concentrate on the graph in the middle here for the moment. What he's got is there's his control group, autotrophic plants, and you can see they have the arbitrary 0 amount of carbon-14 present in them. Okay, because these plants have fixed their carbon recently. In other words, at this end of the graph for the carbon-14 distribution graph, right? So recent photosynthesis means that your carbon does not have much C14 in it. So that's what you've got there. And for the ordinary ectomycorrhizal plants, that's these ones up here, Monotropastrum, Pyrola, both of these are, well, that one's green, that one's chlorophyll-less, bird's nest orchid, Encephalanthera, and the lecum orchids. All of these, beg pardon, all of these are here. They also have recently fixed carbon in them. Recently fixed because they've been mycorrhizally attached to the autotrophs and have parasitizes. They've stolen the carbon from the autotrophs. And the carbon they've stolen has been recently fixed. So this is the standard story that we had up until the chap looked at these orchid genera here and discovered some weird mycorrhizal associates, which in fact turned out to be saprophytic fungi. And if you look at those, they plot up here. So in other words, these fungi have got elevated levels of carbon-14. And those elevated levels of carbon-14 have been stolen by the plants which are parasitizing them. So those orchid species, including Gastrode, which you've illustrated over here on the left, these have parasitized saprophytic fungi to get their carbon. So we've got an additional kind of relationship here. Instead of a living tree providing the source of carbon, we've got a dead tree providing the source of carbon. That's an armillaria which takes up the carbon, and then it's robbed by the saprophytic orchid. Sorry, it's robbed by the orchid. It's still a mycoheterotroph, it's still parasitic, but it's parasitic on a saprophytic fungus, not a typical mycorrhizal fungus. So this whole business of mycorrhizae is going to get a lot more complicated as time progresses and people do more and more observations. Okay, coming towards the end now. If you want a sort of step-by-step guide as to how mycoheterotrophy actually evolves, you can look at this little diagram here. So it's kind of a little evolutionary tree in a way. So you start out with autotrophy as the basal branch. This is the sort of where you start off. And then you get a branch where you get an initial mycoheterotrophy. So you get a relationship between a germinating seed and the fungus in the soil. There's some carbon gain in the seedling. So you get a bit of mycoheterotrophy going on here in the seedling. But by the time the plant gets to be an adult, it doesn't need the support of the fungus. And then typically what we see is carbon gain in the adult. In other words, where although the plant is green, it is nevertheless still getting carbon from its mycorrhizal associate. And critical to this step seems to be a switch to an ectomycorrhizal relationship. In other words, whereas before we had the ericoid type or the arbutoid type, the ericoid type is an internal relationship, as the mycorrhizae are inside the cells, ditto with the orchids that are inside the cell. You get this switch to an ectomycorrhizal relationship. And at this point, it seems that the adult plants get a carbon gain and you get a partial mycoheterotrophic relationship. And then with loss of photosynthesis, you get full mycoheterotrophy. That seems to be the sequence of events. But what is the driver behind that sequence of events? Why does it happen? So in other words, what are the natural selective forces behind that? Now, one thing I've not mentioned so far, and might well be significant, is that all mycoheterotrophs have got what are called dust seeds. And is this significant? Dust seeds, illustrated top right here, this is Hypophytis monotropa. Basically, you've got a tiny, tiny little undifferentiated embryo. There's no endosperm, and there's a seed coat which is drawn out into sort of two little wings like that. The whole thing is very, very tiny, which gives rise to the name dust seeds. And these things are normally regarded as an adaptation to dispersibility. These little wing-like appendages are believed to allow the thing to be transported through the air more readily. Well, it's relatively easy to believe that might be the case for meadow orchids and some orobanks which live in the open. You can see that being wind dispersed in the open could well be an advantage. You could be at an advantage if you've got very, very small seeds. But in heavily shaded woodland, such as the place where Isabel was exploring out near Melton, it's arguable as to whether wind dispersal is of any real significance at all. And in fact, there are experiments on Pyrola chlorantha in Sweden, where it's been found that 82% of the seeds disperse only 1 meter. 95% go only up to 5 meters. So if this result can be generalized, it's clear that wind dispersal does not have much impact in a heavily shaded woodland environment. And in Japan, where they've looked at dispersal vectors of these mycoheterotrophic species, it turns out that these things are camel crickets, and we don't have those in Britain, but they do in Japan. Camel crickets disperse pyrola seeds there. Camel crickets are a kind of flightless grasshopper, so these don't fly through the air either. So not only doesn't the wind take the seeds, this grasshopper is unlikely to take the seeds through the air either, and it'll disperse them relatively locally to the source population. The Japanese have also found that various species of ant will disperse the seeds. Again, it's unlikely that the ants will take them terribly far. So having this kind of morphology, dubious value in terms of it being an adaptation to dispersal in a woodland habitat. Probably better would be to focus on fecundity as a driving force. This is my last slide, so bear with me till I've just finished this one. The idea is that as the angiosperms evolved in the Cretaceous period and rose to dominance, they created new ecosystems which had not been seen before. So we got the development of closed angiosperm-dominated forests. Admittedly, we'd had closed forests before. You've only got to think of the coal measures. But angiosperm-dominated tropical or closed forests, heathlands and grasslands, successively more recent types of ecosystem which had never existed before. Now, in that context, there could well be selection for high levels of fecundity, in other words, high seed output in some lineages, in order to capitalize on these new ecosystems, finding available niches in them. So in other words, you've got probably a number versus size trade-off going on in different groups. In some groups, there will be selective advantage to having large few large seeds. In other groups, the reverse will be an advantage, a lot of small seeds. And the idea is that these mycoheterotrophs are a necessary a necessary consequence of the selection pressure favoring large numbers of small seeds. Because taken to an extreme, your seed side can get so small, in other words, no endosperm, just an undifferentiated embryo, that germination or establishment becomes impossible without any help. So it meant that there was a tremendous advantage to establishing a connection with a helping hand fungus to get these things going. And this is where mycoheterotrophy developed. And that's how it developed. This is the most recent idea, and it does seem to be quite an attractive proposition. Difficult to demonstrate and prove, but nevertheless, it makes a reasonable story. And my parting shot is it's mycoheterotrophy, not saprophyte. So, are there any saprophytes in the British flora? No. And I'm happy to take any questions.
Thank you, Richard. If we maybe stop your screen share and turn your camera back on.
I'm amazed it worked without the camera, actually.
Well, we didn't get the benefit of seeing you, but we could hear you and we could see your slides. So that was a fair trade-off, I think. So if you're happy to take some questions.
You're still awake? I haven't done a Zoom one like that before.
They're still there, you can see them.
I normally like to see my audience and prompt them with questions from time to time.
There are various ways people can ask questions. You can either put your hand up in the chat window. You can on the side panel, you can type it in the chat window, or if you actually want to wave at me or put your hand up on the video, I'll try and get you a question. Again, we don't want everyone's microphones open at the same time because it will cause us some problems. So do we have any immediate? Yeah, sorry, there was a question there. Was that Ian? Did you have a question? You'll need to unmute your microphone.
Can you hear me now?
Yeah, we can.
Yes, can hear you, Ian.
A question for Richard concerning that dispersal slide. It was, the figures seem to be a funny way around, that it was 95% nearer and 82% nearer and 95% further away. I don't know if Richard can go back to that.
It's cumulative. So 95% of the seeds went up to 5 meters.
Yes.
But 80% went up to 1 meter.
That's less. That seems the wrong way.
No, you add another 15. You add another 15%, which can go 5 meters.
82 closer, 95 further away. Thank you very much. That's fine.
So it's a cumulative total.
Yeah, fine.
The main point, of course, is that hardly any of them go any distance, which is surprising.
Any further questions?
Yes.
Oh, Anthony, yes.
Yes. I'm sorry, there's no picture. Hello, Richard.
Hi, Anthony.
I was thinking about regarding the size of seeds and dispersal. It would seem logical to me if that's plants that disperse over a long distance using fine seeds ought to be trees. I'm thinking of the range of pollen which has been investigated by archaeologists. Pollen of plants growing near the ground, like bluebells, get about far as 6 feet, whereas birch pollen is found at lowest density directly under the birch tree, and it travels much further.
Yeah.
So it seems logical to me that low-growing plants, such as the ones we've been talking about, would have small pollen.
Won't go very far.
Small,
Yeah.
small seeds, yes.
Yes. Yeah. I mean, with trees, of course, the whole business of seed size is wrapped up with establishment, germination and establishment. If you've got nice big chunky seeds that can muscle their way into a closed environment, the bigger the seed, the more packed endosperm and nutrition, the better it will be, better its chances will be at establishing. You can take the alternative route, is to get out of there, find somewhere else to live. So dispersal is traveling far is a key thing to be able to do. But whatever, you do need to establish, and whether you can do that by being a big seed or whether you can do it by being a little seed with the help of a fungus, that seems to be the sort of the general context in which we should consider these things.
Yes, I can see that seeds dispersing over a short distance are more likely to encounter a cooperative fungus than one traveling further afield.
Well, yeah, it's debatable. This is all armchair stuff, isn't it? That's the trouble. We don't actually know.
Okay, thanks. Does anyone else want to ask a question?
Yeah.
Hi, Ian.
What about the brassicas? Because they're not supposed to have mycorrhizae.
About half of them do, half of them don't. So some brassicas do have, some members of the family do have mycorrhizae.
Ah, thank you.
But they are a family. I've seen that as well, saying in Brassicaceae don't. That's not true. If you just bear with me, I can probably give you. No, I can't. I can. It's about 50% which do, 50% which don't.
Ah,
from memory.
thank you. Excuse me. Let me just put somebody's phone.
But there are families which apparently have not had any mycorrhizae recorded in them before at all, and some of those are aquatics.
Ian, did you have another question? You have to unmute your microphone, Ian.
Russell does.
What about that?
Yeah.
What?
Can you hear me?
Yes.
Just quickly, dust seeds, like orchid seeds, is anything known about where they acquire their fungal partner? I mean, what sort of distance or anything like that, and how soon? Very soon, presumably after they hit the ground.
I don't know the answer to that, Ian. I mean, obviously the fungal partner is in the soil, and if the seed lands in a part of the soil without the fungus, they will struggle.
Yeah.
And probably not produce anything. On the other hand, these fungi are pretty widely dispersed, pretty widespread. So
yeah.
I don't know the answer to that, how the fungus finds its orchid and vice versa.
Okay, thank you.
Russell, did you want to ask a question?
Can I just go back a bit? Have I got this right? Are there any plants which are saprophytic?
No.
Can I have a question?
No, you can abandon the term. It's a myth.
Yeah.
Talk about mycoheterotrophs. They're parasites, basically. That's what they are.
Can I ask a question here, Alan? Okay. Yeah. There's I sort of read various botany books and so on, and there's quite a lot of talk in some of the books I've read about trees in a woodland. First of all, I dropped out for about 5 minutes, so somebody else might have said this, that the line just went. But what I was going to say was there's quite a lot of talk about, as you can imagine, a tree is connected to a fungus and another plant, but it might also be connected to another plant and another plant and another tree. And there seems to be quite a lot of discussion about trees communicating with one another via the mycorrhizal roots.
That's right.
And actually, like, say, one tree attacked by a beetle and can send some sort of hormonal messaging through to another tree. I mean, do you feel that that is reasonable? I mean,
think that's been demonstrated. Yes, I think that's right.
So quite some of this new information that's all being built up between the relationships between different plant species will eventually have to be taken into account when we're looking at agricultural, forestry practices, conservation and so on, because it's not just looking at, you know, we want a few trees here and a few plants there. You have to look at everything as a whole, roughly.
That's right. I mean, I think the good thing is that most of these mycorrhizal fungi, I mean, Jeffrey, correct me if I'm wrong, but they seem to be relatively non-specific and so widespread that, you know, the necessary connections establish themselves as soon as you almost as soon as you put a plant in the ground. Yeah,
thank you. Yeah.
It makes it a bit weird when you go walking in the countryside.
Yes.
Thinking, oh my God, this lot's all connected.
Yes, I know.
They're not doing anything wrong.
Yeah. I think Jeffrey wants to ask a question.
Go ahead, Jeffrey.
Mute. Microphone, Jeffrey.
Well, first of all, I'll talk about Richard's point about the fungi. I have, as you might expect, tried to correlate the records of trichiloma in Leicestershire with these populations. Unfortunately, the fungi study group have a number of fairly regular sites that they go to, and they don't go to many other sites. So I don't have any records for Kesson Quarry woods, where the other population of monotrope hydrophytes is in Rutland. I don't have any records for Astleby, but there were some records of the trichilomas in Cloud Wood. Cloud Wood has Meltia nidus avis, the bird's nest orchid. So that is a potential connection. It's only a correlation, obviously, but at least we know it's there. Yes. So there is a connection, but nothing so far with the yellow bird's nest orchid or the Dutchman's pipe. So there is something there to go on. And again, it's quite an exciting research project. If we could get fungi study group to go out to Rutland,
absolutely, yeah.
We might be able to find some trichilomas there and see what there is.
Excuse my ignorance, but what does a trichiloma look like?
It's called a knight. Well, it looks a bit like an ordinary mushroom. It's got nothing particularly striking about it apart from the structure of the gills. So it's not like a puffball or an earth star or something like that. They're mostly grey mushrooms.
Okay. Undistinguished, except for the fact that
I mean, they've got their own special characters, but there's nothing spectacular about them. You know.
Right.
So it would be nice to go and find out whether we can find anything else out there one day. But what I was going to say was going to move on about the dispersal problem. I wondered if you'd had any thoughts about how these populations got there, because this is absolutely extraordinary botanically that these two populations suddenly pop up.
Well, I have an idea which needs to be tested, and that is these dust seeds, I think, could be electrostatically attachable to things like dunnocks or blackbirds, which sort of rummage around on the ground and then fly off with them. But I've no idea whether that's, whether that could happen. Yeah. But it's an experiment we could try. We should get some dunnock feathers and blackbird feathers and see whether the little seeds jump up like feathers.
Right. Oh, that's very interesting. Yeah.
I'm just trying to think, what is going to be on the ground that, brushing against plants which are about 5 centimeters, 8 centimeters tall, and will take the seeds. Nothing's going to eat them, I don't think. I've no idea about any invertebrates. I mean, these Japanese have turned up an ant and one of these strange cave cricket things, camel crickets. But it's a long-term job, I think. You've got to sort of sit there and wait.
Yeah. I mean, you've
See
seen
what happens.
yeah, I mean, you've seen the populations. How long do you think they've been there?
Goodness only knows. I mean, they must postdate the abandonment of the island.
Yeah.
So we can put an upper limit.
Yeah.
But it's only going to be decades, isn't it?
Yeah, and it's astonishing. I think it's absolutely astonishing.
But these post-industrial sites, which Hazel mentioned them earlier, I think, they do seem to be a magnet of dust seeds. So, I mean, epipactices in Glasgow colonized the old factory sites like that, like nobody's business.
Yeah.
So, you know, clearly the dispersal is an element. I don't think you're going to get anything other than wind is going to take epipactis to an abandoned industrial site in Glasgow. You sort of think there has to be wind, but on the other hand, yeah, no, it's a question of where the source population is. If it's in the open, maybe you could believe it, but if it's in a closed woodland, harder to believe that.
Yeah, fine, thanks.
So I don't know, Jeffrey, sorry.
Okay, at this point then, I think I'm going to formally thank Richard for his presentation. It's very nice, yeah, well deserved, Richard. It's very nice to have a speaker who answers the question that they've posed in the title of their talk with a question mark, but we've got an answer. So no, there are no saprophytes in the British flora, at least that we know of so far.
That we know of so far, yeah.
We know of so far. So thank you for a very engaging talk, Richard.